In the midst of a European summer, the earth cracks under the sun's relentless rays, an almost barren tract of wasteland, long abandoned by man. Surely most of the life here has withered away. Perhaps there is nothing left to offer. Yet far from being stripped of life, this seemingly inanimate landscape is in fact vivacious and bountiful. A closer look, and we observe life adapting within this hostile stretch of land. Just a few meters away, pioneer plants have established themselves. Stirred from dormancy, seedlings have sprouted from the ground months earlier. In July, the field is teeming with self-sown flowers. Their vibrant colors are woven with the dry blades of grass. Not only do these plants survive, they thrive and transform this arid soil by replenishing and restoring its structure. A wildflower meadow may have an aesthetic value to us. However, their importance far exceeds their beauty. They are the backbone of this ecosystem. These habitats are home to different life forms, from those invisible to our naked eyes, to a variety of insects, different types of birds, and even small mammals. Together they form a self-sustaining yet fragile ecosystem. As we dissect the constituents of these habitats, we find that all life forms are connected. As the naturalist John Muir once noted, when we try to pick out anything by itself, we find it hitched to everything else in the universe. At the peak of the flowering seasons, an assemblage of pollinators can be observed. These naturalized habitats are the principal source of nectar and pollen. Here, a hoverfly busily feeds on a yellow chamomile flower. These hovering specialists are important pollinators of both wild and garden plants. Nearby, a blue-tailed damselfly rests in a raspberry bush, and a bumblebee hovers over the flower heads of bird food trefoils. This ecosystem, left to its own devices, serves as a hunting ground and a shelter for small wildlife. Those that are much smaller than us, but nonetheless indispensable members of the food chain. A large part of temperate habitats is composed of members of the Leguminosae family, commonly known as the pea and bean family. Often found growing in wild habitats are the bird fruit trefoil, Lotus corniculatus. The white clover, Trifolium repens. Its close relative, the red clover, Trifolium praetens. And a hybrid between these two species. Occasionally you may find the crown vetch, Coronilla varia, the hop trefoil, Trifolium campestre, and many other species of legumes in their various forms and colors. But perhaps more familiar members of this family are those popularly grown in gardens, the broad-leaved everlasting pea, Lathyrus latifolius, wisteria, and lupins, to name just a few. Beyond the realms of our gardens, many are highly important food, forage, and biofuel crops, such as alfalfa, Medicago sativa. Green beans, lentils, and chickpeas. Globally, grain legumes contribute to 27% of crop production and are the second most important crop plant after Poaceae, or the grass family. This is not surprising, considering that Leguminosae is the third largest flowering plant family, consisting of almost 800 genera and 20,000 species. In one small field alone, several species coexist. They are highly diverse in terms of morphology, life history, and biogeography. They have radiated into nearly every habitat, from deserts and savannas to tropical forests and Arctic regions. What is it that makes these plants one of the greatest lines of land-plant evolution? How do they become so successful? And what role do they play in agricultural and ecological systems? Perhaps this family's evolutionary triumph owes itself to the fact that many legumes possess the ability to engage in a symbiotic relationship with soil bacteria, collectively known as rhizobia. This close interaction between plant and bacteria is advantageous for both partners. In Legume-Rhizobia symbiosis, bacteria are provided with photosynthetic products derived from the host plant. In return, legumes receive metabolically usable forms of nitrogen, the product of a process termed biological nitrogen fixation, or BNF. By interacting with compatible rhizobia, legumes can develop a highly specialized root structure called the nodule. It is within this protective and nutritious structure that BNF takes place. Within legume nodules, rhizobia converts atmospheric nitrogen into ammonia, which is then used by the plant to form many biomolecules, such as proteins. BNF has an important ecological role. This is particularly true for nutrient-deficient environments, as nitrogen that is fixed by legumes is returned to the soil in forms accessible to other plants. At the same time, soil health can be significantly improved, promoting its capacity to support the microbiota, flora, and the living ecosystem as a whole. Although nitrogen is the most abundant element in our atmosphere, the availability of the metabolically usable forms is strictly governed by the process of nitrogen fixation. It is an essential element for plant growth and productivity. Among all necessary nutrients, it is required by all plants in the largest quantity and is often the most limiting factor for crop yields in agricultural systems. It is hardly surprising then that there is an ever-increasing interest in the application of legumes and BNF to modern crop production. What do nodules actually look like? They often appear as small, cylindrical, or spherical lumps on fine roots. Light microscopy reveals multiple nodule cells being occupied by rhizobia. Nodulation is a highly specialized process whereby a specific group of legumes will only interact with certain rhizobial strains and vice versa. On its present course, agriculture is in urgent need of utilizing available resources in a more efficient and equitable manner. Despite agronomic knowledge and technology available for implementation, issues resulting from land mismanagement and natural resource exhaustion largely prevail. In many regions, the gap between actual and potential yield is widened as a result of inefficient irrigation, nutrient imbalances, and inappropriate practices which lead to soil erosion and degradation. Cultivation of crops on land not suitable for specific species or varieties further leads to an increase in chemical inputs, resulting in large amounts of pollution. Modern high-input agriculture largely contributes to the breakdown of biological, self-sustaining recycling systems. Issues relating to ecological imbalance and agro-industry pollution are intensified as biological systems become increasingly dependent on external, manufactured inputs. Meeting global food demands by means of agricultural extensification and intensification is unsustainable. It has devastating environmental effects and is simply unaffordable for small-scale farmers. With climate change occurring at an unprecedented speed and global population projected to reach 10 billion by 2050, agriculture must not only be able to increase in productivity, but also shift to more sustainable practices. One method of achieving a more sustainable agricultural system is the exploitation and enhancement of BNF via Legume-Rhizobia symbiosis. However, the use of legumes in raising productivity in farming systems has largely declined, partly due to new technologies made available during the Industrial Revolution. Over the last century, there has been an increase in the application of synthetic nitrogen fertilizer produced by the high-energy Haber-Bosch process. An excess of inorganic nitrogen is detrimental to soil and water by depleting the soil of carbon, reducing soil moisture content, and contaminating water. Ironically, excess nitrogen fertilizer also depletes existing soil nitrogen by promoting microbial utilization of carbon, resulting in the net loss of soil organic matter. Consequently, the soil's capacity to store organic nitrogen is reduced, and large amounts of nitrogen is leached from the soil. In recent years, researchers have given priority to searching for potent microbial systems with the aim of improving agricultural productivity in a highly efficient and sustainable manner. Our research aims to investigate the nodulation status of tropical legumes, with a focus on those growing in the wilderness of the northern and northeastern regions of Thailand. The tropics and subtropics harbour the greatest variety of legumes, and therefore of potential nodulating rhizobial species. Following current nodulation records, we will investigate all three subfamilies of Leguminosae. From obtained nodule samples, rhizobial isolates will be identified and authenticated on the host plant. The efficiency of nitrogen fixation can then be determined for each Legume-Rhizobia partner. In addition, rhizobial isolates will be used to inoculate non-host legumes. In other words, different rhizobial species will be used to infect the roots of agriculturally important legumes, and their nitrogen-fixing efficiency will be subsequently assessed. This project could allow us to provide agriculture with new legumes and rhizobia to directly enhance pulse yields. Also, we may be able to employ legumes not currently cultivated to transform infertile and nutrient-poor soils in agricultural and ecological systems. Our findings will form the foundation for further research into the specificity and efficiency of legume-rhizobial partners, particularly in Thailand, where comprehensive reports are limited. With the scarcity of natural resources in mind, our understanding and application of this specific symbiotic relationship will no doubt play a vital role in future agriculture and ecology. In an undisturbed meadow, we recollect our thoughts in tranquility amid thousands of flowers. The stories told by this seemingly silent world capture our imagination and enrich our lives. The closer we look, the more intricate these stories reveal themselves to be, often presenting new and unsolved mysteries. The quiet members of this wild habitat continue to play a significant role in our domesticated lives.